Welcome to this episode of Eureka. Let me tell you one comedy part of a film that I saw when I was a college kid. It was in a Tamil movie. One homemaker was listening to radio and she had put it on one station. But that radio was an old radio, so that station was shifting from one station to another station. In the first station, they were running a program on how to take care of a baby, young baby. In the second station, it was how to make a lemon pickle. Okay, so the program started with the first station where they were saying that wash the baby properly. Okay, by the time the station shifted and went to another station, and then there it was saying that cut it into small pieces. Okay, then it came back to the first station and said that apply powder evenly. And it came again to the second station and said that have as much as chili you want to put. I am sure it was very hilarious, when one station is talking about taking care of a baby and the second station is talking about making lemon pickle, and if you mix it too. It's a crosstalk. What happens in our body? There are billions and billions of cells, and then they are responding to external stimuli. What if there was a crosstalk? The guest we have today, Dr. Soumen Basak, is working on this issue. We are going to watch a very interesting conversation with him, but before that, we will take a quick look at his brief profile. Keep watching Eureka. This will be a very, very interesting conversation.
Crosstalk, the process inside a cell that occurs when the same signal is shared by two or more signaling pathways, and investigating such pathways of crosstalk can help in understanding many diseases. It is where Dr. Soumen Basak has extensively worked, presently the head of the Systems Immunology Laboratory at the National Institute of Immunology, New Delhi. Dr. Soumen Basak carried out his doctoral studies from the Biochemistry Department of Calcutta University. As a PhD student, he elucidated the molecular mechanism underlying gene expressions by Chandipura virus, an emergent human pathogen. In 2003, he joined the laboratory of Professor Alexander Hoffman at University of California, San Diego as a postdoctoral fellow. During his postdoctoral tenure, Dr. Basak began to use systems biology approaches for deciphering complex cell signaling mechanisms. He joined the National Institute of Immunology in 2010 and set up the Systems Immunology Laboratory using funding support from the Wellcome Trust DBT India Alliance. Dr. Basak's group has been addressing how intricate immune signaling mechanisms, particularly those involving the NF-κB system, modulate inflammation in physiological settings, and how deregulated cell signaling pathways trigger aberrant inflammation in human ailments. Dr. Soumen Basak has recently received the prestigious National Bioscience Award for Career Development from the Department of Biotechnology, Government of India, and the Shanti Swaroop Bhatnagar Award from the CSIR for Research Excellence in Biological Sciences.
Thank you for being with us. Let me start with one simple question. See, in a typewriter or in a computer keyboard, when I press A, I get a response in the screen, my A comes. Okay. So, if I am a cell, it's like a computer. Okay. So, there is a external stimuli, let's say dust. Whether it is my eye or whether it is my kidney, it should produce the same A, right? You are saying it does not.
It does not.
That's the complexity.
That's the beauty of and complexity of biology. That's where it is that it always integrates multiple things together. It's again not only about kidney and eye context of eye, but it is also about let's say a bacteria versus a virus. You are, even for a kidney cell or maybe a cell macrophage that I will probably say, they will respond differently. That's the beauty, that you have all these different, nice curated different things, and you are able to recognize and respond differently. And we have been focused on to address this, that how such a, as I said, permutation and combination, how you generate such a diversified responses, calibrate them. Again, analogy sometimes helps. If you remember of those beautiful concerts you might have visited in this evening, that guy is beating a drum, you get a sound. That guy is hitting a piano key and you get a sound. And that is the composer mixing this sound to different extent and length, and you get different kind of music comes up.
Okay.
So we are talking about, even for a pathogen, it's not always about one pathway that they are activating in the cell. These are immune signaling pathways. They are activating certain immune signaling pathways. There are these microenvironmental signaling pathways, right? And then there is another pathway that is activating some other signaling pathways. But this bunch of pathways, this cohort of pathways, their combinatorial activity and how strength of each of these pathways or signals eventually will lead to a particular cellular response. So it's not one pathway that you say typewriter A, typewriter B. It's never like that. It's always multiple things together, and how much extent of mixing of these signals.
And then that is the where the music comes.
That is the where the music comes, and it is with the same drum and same piano. I can give you so many different musics. We agree, right? So this is what biology knew before mathematicians we did, right? So nature knew. So, and that is why they are doing it, is because that, as I was articulating to you, then potential threat is infinite.
See, suppose if let's say there is a linear connection. If I pick up the phone here, this side the phone rings, right? Then I can know that this is what is causing that, and the cause and effect relationship is easy to establish. When there are going to be multiple inputs, and then your results are also going to be very complex, how do we know that because of this that is happening?
Yeah, this is a very excellent question. So when you have this sort of thing, that multiple signals, and you're talking about potentially engagement of them together to different extent, and as you have articulated, it generates non-linearity, and this is where you will have non-intuitive properties. It's not going to be the same one cause and effect anymore, and there are ways to probe them, and one advancement has been in last decade or two is the advancement of systems biology that recognizes these complexities, that recognizes this non-linear behavior of biology, biological system. So what you can do, you can actually in insulation, you can study the cellular pathways, pathway 1, pathway 2, and then you can also rewire these pathways in computational model. Once you model them, you can play around with it. You can find out if these pathways are engaging with each other in some sort of crosstalk, right? That's the key word, and thereby can generate what kind of possible responses that cell can generate. You can, and then from those you can direct experiments for a select set of scenarios and test them if they are happening in reality, and then again use model and experiment going back and forth to really dissect, tease out the key mechanisms, tease out this key component that generate this sort of non-linearity, and then try to envision that how this might impact biological system, let's say immune response by macrophage or gut intestinal cells that we are very interested in. So this is one model which we are with the systems biology and interdisciplinary science. We have been addressing this sort of complex system where it's not about one pathway or two, but multiple pathways. Yeah.
So you had talked about two parts in it. One is the actual experiments carried out in wet labs like this, and the second is computer simulation. So let's take the wet lab scenario. In a wet lab scenario, what do you actually do? For example, if in a very, very simple way, for example, we understood scurvy is because of lack of vitamin C, is a very simple experiment. A group of scurvy affected people, some of them were given lemon, some of them were given different things, and then they found out that people who were given lemon or citrus fruit or fruits with rich in vitamin C cured, others did not cure, so they could understand easily a connection, right? So in a wet lab, what do you actually do? How do you actually conduct these experiments? What are the methodologies that you use?
Yes, so let's talk about immune cell. Take it out of your body, put it in a laboratory settings, and then hit them with something that we call pathogen associated molecular pattern. They are associated with pathogens. We have purified component from those pathogens. You hit those cells and then study in the cell all these signaling pathways that actually transmit these pathogen signals. how they are activated, how they are inactivated, what are the temporal dynamics, kinetics, and all this. What kind of genes, expressions are occurring because cells are now exposed to pathogen signals. And then you can do combinatorially, not only pathogen signals, but you remember that eye and kidney has different microenvironment. What kind of signals do they get if they are in those microenvironment? Can you, in addition to pathogen signals, add those signals and study if something unexpected happening because signals are together just because of that, and try to rewrite those in mathematical model. You generate a conceptual framework, why is it happening, and exactly what are the non-linear areas one should be focusing on, and what kind of other thing one might expect in addition to what you are already seeing. So it opens up. I will remind you about those Apollo missions, right? We landed up in moon, and we did it pretty good. In 1960s we are talking about. Why did it we so early is because we had the computational model, we had this physics, we had this law, we had hundreds and thousands of simulations before we landed on moon. Biology, we need this kind of computational tool with predictive power. That's the thing that we are very interested. We are really putting enough effort into it.
Coming back to the wet lab thing, one of the things that I saw in some of your research work is that you use something called knockdown method. So knockout method. So what do you do? I mean, you take a cell and then knock it out?
So there are knockouts. That means that you have, let's say, some gene 1 that is very important for pathogen response, or gene 2 that is one that transmit this kidney signals or microenvironmental signals. So you want to really now dissect it, right? How do you dissect it? Now you want a cell where you can target it way. I mean, we know it's not really from cell. We start from animal, basically generate an animal where this gene has been disrupted, and then those knockout mice. What in our laboratory we do, we collect cells from those knockout mice. And then test the function of this gene A that transmit pathogen signal, or function of this gene B that transmit microenvironmental signal in overall cellular response that we are basically interested in, and that has ramification for inflammatory.
So that's one methodology that is used.
Knockout.
The other methodology is something like chemical methods, or something that you also use, right? Can you explain that?
Yeah, so that we are doing it these days. It's beyond this, laboratory cell that I said, that I isolate cells from a body and study that, and then you have computational model. But these all generate very powerful hypotheses. Eventually, people will say that what happens in body, and that is what we sort of began to use some experiments in even animals. We have methods to induce intestinal inflammation. That's probably we are referring to, that you can have chemicals that can cause intestinal inflammation, where intestinal cell will be exposed to. We have a lot of pathogen microbes in our body. Cells will be exposed to those, and you can study the responses. That is it matching up to what you have been thinking about, combining your cells and computational models. If it doesn't, most often it doesn't, by the way. It gives a great deal of learning for next few years. There must be some systemic areas which you have to rectify, and then make a superior model. This is all about never-ending loop. You refine your model, go back to your experiment, test, and then some works, some doesn't. Then say that why that the model gives you an idea that there will be areas which can improve.
Okay, so this is about the wet lab. Now coming to your other part, which is actually using computers.
Yes.
So when you say we simulate, it's like a black box. What do you actually do? I mean, when you are going to make computer into a simulative environment, you have to make it behave like a cell. See, if I'm going to program a computer to behave like a car, so that, for example, I can learn driving. I mean, I know how a car works because I know physics, right? So I can easily make a computer into this. But here you are venturing into something unknown.
Very good point.
So, so how do you code something which is unknown? That's a big mystery, right?
That's a very good point. That's a very good point, because the analogy was strong. When it comes to car, the laws of physics are so defined. The laws of biology are not. So, and in my lab, we are always, as I was trying to tell you, that computational model experiments, they are going hand by hand. But in computational model, to really enlighten your black box, we are not modeling the entire cell. That's the first point.
That's the important point.
That's
so
very
the computer
important.
Doesn't behave like a cell.
It's not a virtual cell.
Okay.
That's the first point, because in biology you need to limit your scope, because there are a lot of unknowns, and that you can possibly model with some degree of confidence is things that are known to you. some pathway A and pathway B, I know quite deal of it.
Okay.
Why not model it? I know there are 200 other pathways, but
And then combine them together.
that's the bottom-up approach. You are getting it. So I got pathway A, pathway B. I'm modeling it. I know all the molecular details. I'm putting it in and then simulating them together sequentially, simultaneously with different strength and different crosstalk, and see what kind of responses I can get. And trying to see is this explains some physiological scenario I was actually some day back I encountered. That's one thing. But then we know that this pathway and B is not sufficient. There are pathway C. And as you encounter new problem, you add pathway C in your model. That's called bottom-up system biology. You start from things that you know, and from bottom up, you try to go to this whole system, which is enormously complex.
That's an interesting part, and perhaps this is where we need to take a very short break. Keep watching Eureka. This conversation will continue after a very short break.
Which article defines qualification for the membership of Rajya Sabha? Article 84 of the Constitution lays down the qualifications for the membership of the Rajya Sabha.
Welcome back to Eureka. We are having a very wonderful, enlightening conversation with Dr. Soumen Basak. Before the break, we were kind of talking about your systems biology, in particular systems immunology in general, to understand the lay of the land kind of stuff. Now, let me come to a specific area that you are working. One of the pathways that you are working is called as NF-kappa B, right? So, why it is named like that? Why it is NF-kappa B? It seems something like a secret code. What's it actually?
So these are transcription factors, a family of transcription factors was originally identified from B cells.
Okay.
These factors were supposed to, they were identified as a transcription factor mediating Ig-kappa light chain gene expression, and thus named nuclear factor coming from Ig-kappa. So kappa of B cells. Okay. Eventually, later we found that this is almost present in every cells, particularly very important for immune cells. And from 2000 onward, we know that this is one of the most important factor for cancer cell survival. Okay. So it's not only immunology but also in cancer. And this is with this very core interest of my laboratory where expertises are. And I should inform you that this is historically this field has also been immensely benefited by scientists of Indian origin.
You mean to say this whole kappa B, NF-kappa B, there is a lot of Indian hand in that? Huh?
This is a very important research area, as you know. It has importance in immunology. A lot of drug trials has been performed in cancer biology, but original discovery was made by Ranjan Sen, who is an alumni of IIT Kanpur. He was doing postdoctoral research with Professor David Baltimore, who is a Nobel laureate. And this is when he discovered this factor. Later, Professor Shankar Ghosh, who is currently chair of the Columbia University Immunology Department, he made tremendous contribution in those early days in 1990s for us to understand the possible function and mechanism. Okay. And then there is this Professor Gauri Ghosh, who is also an alumnus of Calcutta University, who is currently professor in the Chemistry and Biochemistry Department, University of California, San Diego. He helped us to understand the structure of these factors, how then it functions in mediate gene expressions and so on and so forth. So it has always, and there are many more to limit our discussion, but these are the three primary individual.
Okay. And all of them are Indian born.
Interestingly, of Indian origin scientists, and in that way I sort of take some immense pride that being an Indian origin scientist, albeit doing research in India on NF-kappa B research, and that's a extraordinary message. And in this laboratory has been visited by all three that I have named. They have all, and we are very good in touch with them. They have done enough interesting mentoring for us, and it's really privileged that in India, sitting in India, we can do those cutting edge research, and to connect it to your crosstalk, right? When you're talking
so
about
where does this NF-kappa B come into your crosstalk?
So then you remember that we talked about your stone or little dirt in your eye or in kidney or a pathogen, and then you talked about pathway A. That's your NF-kappa B.
Okay.
And so this is always, I mean, there is no exception to this rule. It's an evolutionary concept starting from Drosophila. you have pathogen response requires NF-kappa B activation, right? And then we have all these other signals we are talking about, microenvironments, so on and so forth, anatomic niche derived signals. They do also, albeit differently, modulate NF-kappa B function. But when they are together, what we discovered, this crosstalk can generate cell type specific and stimulus specific tailored NF-kappa B responses. Just because you have two pathways, the second pathway, the microenvironmental pathway, that is in your kidney versus eye, they do not on its own do much. But if they are along with the pathogen responsive pathway A, they change the way NF-kappa B responses are happening, and that's the whole thing we discovered, and we have been working on it more because I think that there are a lot more to be discovered on that.
Then there is one very interesting question. See, whenever we hear crosstalk, okay, it means that there is a communication failure. That's what is our general, everyday experience and perception. But you are saying that crosstalks are actually one way the cells are able to manage multiple inputs that are
and give better output. But you are right that you remember you started with pickle and the baby. So sometimes things goes wrong. That you are supposed to have a very little of signal B and very mostly signal A, and you end up doing opposite. So basically that same very composer can make you leave the auditorium if they compose right. So this is what we talk about. We have found out in multiple myeloma, I mean again NF-kappa B, immunology, of course a bit of cancer, that there could be, I mean we speculate, I mean there are some research we did, more has to be done, but we think that there could be areas where this signal mixing process is defective. And right now we are doing a lot of research in laboratory in intestinal inflammation. That's something related to inflammatory bowel disease. We feel that this is probably for real. We have now human data as well to suggest that maybe the signal mixing process sometimes becomes defective, and you get all sorts of problem. It's not that signal 1 and signal 2 is defective.
It's a mixing is there is a problem.
When you study in isolation signal 1 or signal 2, you get boring accent that this is a disease. I don't see any problem. But you should be thinking how they are mixed.
This is where I come to a very interesting question again about your research and opening up a new field. That till now, most likely, if I have to use the same analogy, people were studying that if I beat a drum, how does the sound come? Somebody is studying that if I play a flute, how does the sound come? That people were studying things in isolation. I suppose your group and some of you are the first people to pioneer of trying to look at multiple inputs.
This has been an extraordinary, I think when I in retrospect think, when I first time said that, people were giving me all kind of look. So in these laboratories, most of the supervisors will tell their student, extremely careful, absolute clean room, clean setups, and activate, take your cells, sterile condition, and add one or the other signal, or and find one or the other response and report back. And I think it has been quite some time, believe around 10-15 years. I first time said this is the most wrong thing to do, because in our physiological setting in nature, as a life scientist, you do not have to be immunologist. It never happens. Cells are always concomitantly or sequentially exposed to multiple things, and that's the real challenge in biology. That is we are investigating here. So if you say that one signal, one response, second signal, second response, but in biology both are together,
completely different.
How you mix them, depending on you can get different kind of responses. That's the strength of immunology, that you can match up to 1,000 different scenarios and calibrate your response.
This where I want to ask you this question. I mean, you have been bestowed with India's highest scientific honor, Shanti Swarup Bhatnagar Award. So when you came to know this information that you have been awarded with this award for your pioneering work in the area of crosstalk, what did you feel? How did you feel that time?
Oh, that was a bit, the few seconds of emotions was, of course, you feel about your immediate family members, right? And people who are not with you, people who are with you, your wife, of course, Manideepa, she is in faculty in IIT Delhi. And my daughter Trisha, there, of course, with us, and it's a very interesting moment. Then your PhD advisor and postdoctoral mentor, Professor Dubajyoti Chandropadhyay, who is Vice Chancellor of Amity University now, Professor Alexander Hoffman in University of California, Los Angeles. These are the people who has helped a lot in your journey. And then we are talking about laboratory full of students. They're tireless. I mean, this is in this laboratory, even in 10 o'clock at Sunday, you will find people, someone is doing something. And their contribution, their colleagues, their help is extraordinary, and you get overwhelmed.
Implication of this kind of work. Okay, of course, to understand and know how the world is, the kind of work that you are doing is very, very helpful. Do they have any other practical implications for drugs or treating diseases? Is there any implications on?
Yes. What we do though, I mean, this is, we are in National Institute of Immunology, right? It's an extraordinary infrastructure where we could do those cell culture animal experiment computational model. And we are within our mandate where basic studies, basic fundamental research in immunology and related areas, it's been mandated. In general, what we do is not really exactly focused on particular disease or cure or microbes, but basic immunology. Right now, we got a very good fitting of our research with inflammatory bowel disease I just articulated. Okay. But in a way, this is a basic research where we find that this can have rather multiple future avenues. I can give you one particular example. That we were talking about pathway 1, right, and that is microbial pathway, and then sometimes you have too much of activity of this pathway, too less, you may have some problem. And we have been trying to make drugs with this addicted mind that pathway 1 is the one, that is the primary, call it in instructive pathway, right? And it is not going to work, and this has been multiple failures by multiple individuals, multiple countries. What happens that these pathways are so instructive, they are involved in so many biological processes. If you block them, you will get side effects. Yeah. Absolutely. Now you got that. Then you think about this pathway B, which is on its own not doing much, but doing crosstalk and generating cell type specific, pathway specific, pathogen specific, disease specific activities. Instead of blocking pathway 1, how about you block the interconnectivity between pathway and pathway B?
That way,
can you get, so this is, I think our vision is that this is the area of systems medicine. We call this is the futuristic therapy. Our research isn't basic, but this appeals to this therapeutic.
These are the implications that is possible in the future.
These are the audience where they can sort of get something and they benefit out of it. That is a fantastic thing.
That's I think a very important point. That of course first we need to understand what is there. Only then we can think of how do I use it. So of course one should certainly do good amount of foundational basic research to go into the next level. That is very, very clear. In this area of study where we are trying to look at how cell response, particularly in immunological sense, to stimuli, particularly pathogens and others, where are we? Are we at the cusp of discovering the almost everything, or we are just only at the beginning stage?
This is a good question. When the problem here is that enormous complexity of biology. We sort of sometimes we have these systems. This is, I was talking about top-down systems biology. So that has been an explosion of information for us. So we can tell you about almost every gene in a cell, and what are the expression level, every protein. This is only problem is with this information. Then you stare at them, and they stare at you. It's a huge table of numbers, and you have no idea what they mean. And that's the challenge that. Technically we have advanced, we sort of can give you everything you need, right? I can throw a dictionary on you, but if you do not see any sentence on them, it is not meaning.
So we are just at the beginning.
Bottom up is going to really probably, it is very challenging. I do not see that in next 5 to 10 years or 20 years we will advance to some extent. There will be a remarkable progress, but maybe it will allow us to construct sentence out of this dictionary. With some methodology, maybe bottom-up systems biology, or maybe something new will evolve to have some degree of understanding.
That's I think a very interesting point. With this, we need to end this conversation. There are many things we can talk, but for want of time, keep watching Eureka. We'll come back with another interesting conversation next week, same time.